Hi everyone, this is Miss Polipchek. I'm going to go ahead and go through our start of notes. So this is Lesson 1, Nucleic Acids. And if we flash back to Unit 2, so in Unit 2 we talked about the main macromolecules, and I just briefly introduced nucleic acids, and we didn't really go into too much detail other than the elements that comprise it, the monomer. So I'm going to do a little bit of a review here and then add some things to that. So our next unit is DNA, RNA, protein synthesis. So we're going to have a whole unit on that content. And with everything in the news, this might be a little bit more interesting to you as well, because viruses tie in here as well. So when we talk about nucleic acids, and we talk about a virus, so a virus has an actual protein code with a membrane, and inside it either contains DNA and RNA. And so it has to have a host to rely on, but with that, it will either inject its RNA or DNA into a host and allow it to produce a protein, some kind of viral protein or proteins that it needs. And so because of that, it's able to thrive and survive and have quite a variety of hosts, which is why they are so successful in what they do. Now, automatically, we think of bad things with this, but there's a lot of really great biotech that we use this process for too. So we're also virus users as far as when we want them to get them to do things that we want. So we'll kind of have a side lesson or lessons on some biotechnology to get into that as well. But I'll go ahead and get started. I'm getting off topic now. And let's look at nucleic acids. So nucleic acids are information storage. So that's their whole goal. And so if you look at this blueprint for building proteins, you can say DNA, RNA, proteins. Well, DNA is our blueprint for everything. So in other words, everything you need to make you is in your DNA. Whereas RNA is very focused and particular. In other words, RNA wants to make specific proteins. So maybe I'm an RNA strand that wants to make cellulose in a plant for some cell walls. So RNA will actually be created from DNA and read that blueprint of just the instructions to make cellulose, and then make cellulose, put that out into the cell. So RNA is going to get specific sequences from our DNA to create those proteins. So that's kind of where we're at level-wise when we talk about this genetic material. So we have our two ideas then. We want to transfer. We've already talked about DNA replication just very briefly. Remember, that happens in the S phase of synthesis. We want to transfer that information that our DNA has to all our cells. We want everything to have that blueprint. And then we also want RNA to be able to be created in all those cells too, because remember, we're going to have specific proteins that we need to make depending on the type of cell that we are. And it could even be that we're talking about specific phenotypes that we have as well. All right, so just a picture of a DNA strand. You can see that twisting, and we also see some letters in the middle there, that A's, C's, T's, and G's, and we're going to talk about those shortly. And remember that that's going to condense very tightly around specific proteins to create our chromosomes. We talked previously in last class that chromosomes have genes on them. Genes are made up of alleles, which are different versions of that gene. And so that's kind of where we're at now. I'm going to upload some videos for you to watch just to kind of go over the basics of DNA. And then, of course, we'll have more notes and discussion on that as well. So we have two main types of nucleic acids. We have RNA and DNA. And you can tell the difference just in the name. So ribonucleic acid, deoxyribonucleic acid. And so deoxyribonucleic acid, DNA has deoxyribosugar, and RNA has ribosugar, ribonucleic acid. So the name kind of tells you. So RNA is really a single strand, but it's going to have the same main components, just a little different versions of that. And DNA is going to have two strands, and they're both going to be in this twisted helical nature. And if we flash back to our structure of proteins and the different levels, you probably are already thinking of maybe some, or hopefully thinking of some of those bonds we talked about previously and how they go together, how they make what they do. So the monomer, in other words, the basic building block of the nucleic acid, is a nucleotide. So remember previously we talked about like a protein is made up of amino acids. The lipid is made up of fatty acid and glycerol. And then what am I missing? Carbohydrates are made up of sugars, and then we'll strand them together to make a polymer. And so here is the nucleotide. Our polymer then would be a nucleic acid. Now the elements that comprise this are carbon, hydrogen, oxygen, nitrogen, and phosphorus. So if you remember when we talked about our previous ones, we had said elements that comprise those. And proteins had carbon, hydrogen, oxygen, nitrogen in that backbone. They could have phosphorus, but they didn't all have phosphorus. Well, here we have all of the nucleic acids will have carbon, oxygen, nitrogen, and phosphorus. Here's a nice picture of an RNA strand. So you can still see that helical single-stranded versus this double-stranded DNA. All right, so what makes up a nucleotide? We have three parts. We've actually already looked at a nucleotide before when we talked about ATP, so adenosine triphosphate, kind of flashing back to this. So that's actually going to be used to create these nucleotides and to strand DNA or RNA together. So pretty cool connection there that we'll be talking about in more detail later on. We've got a nitrogen base. There are 4 different bases that we see. Just one difference between DNA and RNA. So they'll both have A's, G's, and C's. And then that 4th base pair will change depending on whether it's DNA or RNA. They both have a 5-carbon sugar. And just like we talked about before in the past, so the structure of a sugar with that oxygen in the center, we see that pentose sugar. And so how do they differ then? It's just going to be where those R groups are, right? Where those attached side groups would be. And specifically in the sugar, it's where the hydroxides are. So we call this sugar ribose in RNA because of that, and deoxyribose in DNA. And I should have, where are we at? Maybe up at the top? Yeah, there we go. So if you look up here at the top, the difference between deoxyribose and ribose, so we can see that we've got a hydroxide here on both of those. So the difference is just this hydroxide here. So we've got 2 hydroxides here on the 2nd and 3rd carbon. Notice the numbering again, 1, 2, 3. So we start to the right of that oxygen, 1, 2, 3, 4, 5. And so these are going to become really important when we go to strand the DNA. So we want to combine those monomers of nucleotides into a nucleic acid. So you'll see 5, 3 a lot. And all that means is that 5 is at the top, 3 is at the bottom. And so that's showing a direction. So when we make strands, they always go in that direction where they call the 5-3 direction. So we'll talk about that in a second. We also have a phosphate group, PO4, and they connect together. So it's going to combine that phosphate group, that PO4, with the hydroxide. And so we will have dehydration synthesis and hydrolysis, just like we did before. And so they make something called a phosphodiester bond. And so this is going to have 2 ester bonds, which, if you remember, an ester bond was how a fatty acid and glycerol went together. So we see similarities with some of our other macromolecules here. So phosphodiester just means that there's 2 of those connections and the phosphates involved. All right, so I talked about, need some water. I talked about nucleotides, how we have different ones. Well, they're actually into different shapes, so let's talk about those. So if you look to the right, you can see the picture of the purines. The purines are actually double ring structures. And because of the double ring, how they connect in the center. So if we have those 2 strands and they're connecting in the center for DNA, the types of bonds, well, the amount of bonds, I shouldn't say types, the amount of bonds will change, and that's why those things matter. And so purines are specific shapes, and notice that double ring, single ring when we have the pyrimidines. So adenine and guanine are both purines. They are not complementary bases, meaning they will not fit together. And then pyrimidines, we're going to see cytosine, thymine, and uracil. So let me talk a little bit about those. So the pyrimidine, we just see one ring structure. A's go with T's in DNA. So one purine, one pyrimidine, they're going to go together. And then guanine and cytosine go together in DNA. And so, again, purine, pyrimidine. Now, uracil that you see in the bottom, that's only an RNA. And so it replaces thymine in DNA. So in RNA, A's go with U's, but G's still go with C's. So uracil then is a pyrimidine, and it will complement adenine. So what I mean by complement is they will fit together and be able to attract to each other, almost like puzzle pieces if you see like any kind of template of DNA. So I was previously talking about the phosphodiester bond. So when we're looking at that, so remember oxygen's here, 1, 2, 3, right? So 3 is where that hydroxide hangs off. That's where that phosphate is going to be added onto. And then we'll see this kind of stranding. And it's always going to be added to the 3 side, meaning the third carbon side. So we're only going to move in one direction. We can only add to that direction because that's where that hydroxide is there to free and attach to. So if we were in class, I would do the cutouts like we did before. But I can do something like that as we get a little bit more into this with annotate. So when I draw all my pictures, you just have to, you know how these pictures are. All right, and then that nitrogen is going to just hang off that side. And so this is how we get one stranded, but you can very clearly see the purine and pyrimidine shape of those base pairs in the middle. Now the order, the sequence of that, that's what makes us us. And so that will depend on the specific species that we have. And we've seen different amounts and different types of living things, and they don't really know what that means yet. Like, why is there a bunch of A's and T's here? Why is there a bunch of C's and G's in this one? And they're kind of learning and trying to figure out like what's going on there. So again, always add to that 3 side. Now we have a whole set of notes of how that specifically happens. So don't worry about that yet. All right, so here's the bonding that I talked about before. So A's go with T's. And if it was RNA, it would go with a U. Now that, you're probably thinking, why would it go with anything? It's RNA, it's single-stranded. Well, it will have to read the code from DNA, and that's where that comes into play. So it would never read a U from DNA, but it would read an A, and that's how it knows to make a U. So it will always make that complementary partner. And so as long as you remember A's go with T's, G's goes with C's in DNA, it makes it pretty easy to figure out the complementary strand. So in A's and T's, because of the way that they're structured, they actually make 2 hydrogen bonds, and then G's and C's will make 3. Now, if we flash back to our previous section when we've talked about bonding, if you remember, hydrogen is the weakest bond, but it's still a good bond, and there's a lot that we can do with that, meaning they have a nice attraction, so they're going to go together and lock, and we're going to see that helical movement, but that also means that it can be broken, right? We can separate those 2 strands as well, which poses a really great, allows for a really great opportunity for us to copy DNA, which we'll talk about shortly. All right, so here's a picture of the DNA molecule. So you can see how they fit like puzzle pieces, G's with C's, A's with T's. And because of that hydrogen bonding, we can actually unzip them with the help of a specific enzyme. So of course we have enzymes that will do this. And so when would we need to unzip them? Well, of course, when we're going to copy DNA, right? That would be a big thing. So we see those hydrogen bonds between A's and T's. So super important because it allows for us to be able to replicate DNA. So can you imagine we have all these billion letters? If we just had to wing it and copy it all from scratch, oh my goodness, we would have so many problems. So copying DNA replication is the big thing. Now remember, that's S phase of the cell cycle. And so because they're complementary, if we unzip it, then we can go through and just find the complementary piece of each one. And so technically we don't have two new strands of DNA at the end. We actually have an original with a new one to go into each cell. And again, we'll talk about that in more detail later. So if I have one, I can have a template to build the other. So kind of the perfect situation. So remember, why does a cell copy DNA? Well, we have several different reasons, right? One is reproduction through mitosis. It could be because I'm an asexual living thing and I just need to make copies of myself. Or it could be because I just got a cut on my finger and I need to make more skin cells. Also need to copy DNA before I go through, or at the beginning, right before I go through the first round of meiosis. So my chromosomes are ready to separate and eventually become a gamete. And so we have all these opportunities of when DNA needs to be copied. Now, Watson and Crick, we'll talk about them in a little bit because our first main set of notes is going to be about the structure of DNA and how we came about that. But they were really struggling with what's going on with replication? Like, how does our body do this and do this so efficiently? Meaning there are a very small amount of errors that actually happen in this process. And they really researched and really struggled with trying to figure out how that specifically occurred. So I'll skip these just because we're going to learn a little bit about them. Rosalind Franklin was my absolute favorite, by the way, side note. All right, so this is just what I was talking about a little bit earlier. Just as we analyze these, we start to see different amounts. Look at the bottom, parasites have a lot more A's with T's. Why? We don't know. Obviously, if you have 3 hydrogen bonds, that's a little bit more complex of a connection. And so we see that in more higher level organisms. So why? We're trying to figure that out. All right, another side note, I've actually previously talked about this, but hey, look, here's ATP, right? Adenosine triphosphate. And so what is this? It's an adenine nitrogen base, that's your A. It's a pentose sugar, right? And it's phosphate groups 3. But if you remember, how many do we need in DNA? In DNA, we just need 1. So we're actually going to lose those 2 phosphates. That's going to give us energy. And on top of that, we get AMP, which is 1 nucleotide that we can add to our nucleic acids. So really great situation here. That means we can have GTP, which we've talked about that previously, and we can break those phosphates off and get GMP, TTP, UTP, CTP. So same process. They all have 3 phosphates. When we break phosphates off, we get energy. Remember, ATP is the best one though, right? And so not only are we able to get energy, but we can use that energy to strand those nucleotides together. So really great process, really great way to tie in what we've already learned as well. So yet another use, yet another amazing thing for ATP, yet another reason why cellular respiration is so important. So again, AMP, that's our nucleotide. All right, so just to wrap up, just a little refresh on our previous biochemical compounds that we learned about our macromolecules. So carbohydrates, remember their monomer is a monosaccharide or a basic simple sugar. And so again, remember we have the hydroxide with the hydrogen. So we see that same process, our dehydration synthesis, and then remember we add water to break down. So I'm just refreshing because we're going to kind of see all of these come into play in this DNA, RNA, biotech section. And remember the goal of carbohydrates wasn't about storing information, right? It was about getting us quick energy or storing energy. And storage for energy for us was things like glycogen, or in a plant like starch, those more complex processes, whereas our quick energy, the best energy, was that glucose. And so because of that, we can break things down, take them apart, rearrange them. just like we can the nucleotide, right? So that AMP that we just looked at, right? So because we can take ATP down and do that, now we can strand those nucleotides. If you remember the lipid, so I wanted to show this because we see that ester bond here, that ester linkage that we talked about previously. So that is the type of connection that we're talking about when we're stranding the phosphate with the sugar. So the function of lipids, remember, is long-term energy storage. We also talked about membranes, and things that are lipid-based can pass through unregulated. Some of our hormones are lipid-based, and so because of that, we don't have a way to stop or prevent them from coming in. And then we also talked about the phospholipids that were in our cell membrane. So the actual specific structure of the phospholipid as well, with that phosphate head that was negative charged and that fatty acid tail that was nonpolar, right, non-charged. And so because of that, that exhibits a lot of properties as well. And proteins, our basic unit was an amino acid. We could strand amino acids together to make a polypeptide. And remember, we had different levels. We could just have our sequence. Then we had alpha helix, right? We had that helical. Again, we see that those properties of those bonds, like hydrogen bonding, apply to DNA as well. So we see kind of the overlap here between proteins and nucleic acids. And eventually we get that quaternary structure of multiple polypeptides together that all go for a function. And so when we're talking about RNA is getting the code from DNA to make all of those things. So like, how do I make hemoglobin? Oh, it's in the DNA instructions, and my RNA is going to create that for me. And so again, that's our whole unit. And so anytime we need enzymes, anytime we need specific transport structures, signals, really any of the proteins we talked about, we're talking about that process. DNA has the code, RNA getting that code, and then creating the protein from that. And so because of that, this process is really important, right? That's why our body works the way it does. That's why our body looks the way it does. And so we see a lot of connections here. So just to review again, so today our monomer was a nucleotide. Nucleotides go together to make nucleic acids. And then we just have DNA and RNA. There are different types of DNA and RNA that we'll talk about throughout this chapter. In RNA, they have different jobs, and their whole function is to get information and transfer it, and then create, right? When we're talking about RNA, eventually create a protein. All right, well, that is all I have for today. Just an introduction to nucleic acids. Next set of notes is going to be the history of DNA. In other words, I'm going to talk about what on earth, like how did we figure this out? So 1950s, my parents were born. They had no idea what DNA was until the 1950s. So we're talking about very recent in time, and we'll talk about the journey that they had to eventually get to that point. And super interesting because so we've got kind of battle of the scientists that happened, and it was pretty cutthroat and dirty. And so we'll have some interesting side plots. But it was nice talking with you. Lesson 1 is now complete.